Hey everybody, it's Mr. Bliss. This video is going to be kind of a long one. It is on protein synthesis, and it's really important that before you watch this video, you go back and you watch the videos for nucleic acids and on DNA replication, because it's going to be fairly involved. If you are watching this video and are not a student in my class, please recognize that I am not going to talk about every little aspect of protein synthesis. This is kind of meant to be an introduction to the concept. And if you're looking for a really thorough explanation, there are some AP or DP based videos that are out there or textbooks that you can refer to. So let's get started on protein synthesis. I'm going to start with an analogy here. And it's one that I've used in class before. Let's say that I am sitting at home and I realize that I really have a desire to have my grandma's best cookie recipe. They're like literally the best cookies I've ever had. And luckily, that recipe was world famous, so it got written down in this recipe book at the library. So I go to the library, and when I get there, I realize that there's this massive book that contains all of the recipes that are all of the best recipes in the world. But I only need the little one recipe for my grandma's cookies. So all I'm going to do, instead of taking this whole book home because it's too heavy, it's too unwieldy, I'm going to find that recipe in the book, and I'm just going to copy it down onto a small piece of paper like this. And this small piece of paper is light enough, it's able to leave the library, and I can go back home and I can bake those cookies. I can use that recipe as a guide, and I can put it in my oven here. This is my oven, and I can actually make those cookies. Well, obviously this is an analogy. It's kind of a cumbersome one, but that's okay. This is an analogy for the process of protein synthesis. Our cookies here are going to represent proteins, a specific protein that I want the recipe for. I'm a cell, and I want the recipe for a protein. Well, where am I going to find that recipe? It's going to be listed in the massive recipe book that is our DNA here. But our DNA contains all of the recipes, and we only want one. And so what we're going to do is we're going to copy our recipe down. And this is actually going to be the first step in protein synthesis. We're going to call this transcription. And to transcribe something means to copy it, to rewrite it. And so we're going to transcribe it onto this small recipe book here, this small piece of paper. And in this example, in this analogy, this small piece of paper is going to be our RNA, specifically our mRNA, our messenger RNA, which I can walk away with. I can leave where the main recipe book lives, which is the nucleus. I can leave that area. I can then exit the nucleus and go to a place to manufacture our recipe, our cookies in our analogy, our proteins. In this example, our oven, our home is going to be the ribosome, which you may remember from past videos. Ribosomes make proteins. And the actual making of this product from a little recipe is called translation. And this is where we're converting the genetic code into a physical, tangible object. So this idea of taking genetic code in our DNA and turning it into a smaller code into RNA, and then using that to build proteins, that's often referred to as the central dogma of biology. It's one of the most important ideas in all of biology. And it's really how you understand why DNA is so important. It's how you build organisms and how you build cells. So again, this process of going from DNA to RNA, this is transcription. And the process of going from RNA to proteins is translation. And that's what we're going to cover in this video. Simplified, of course. Okay, so the process of transcription. First, it's important to recognize that in a DNA molecule, not every single base codes for something. In fact, most of the DNA is kind of extraneous information that we call introns. And some of it also contains important things known as promoters that kind of guide enzymes or guide the processes of DNA replication or transcription and indicate to the enzymes what to do at those points. Also, sometimes genes or genetic information can be separated by this by these introns. Specifically in transcription, as an enzyme is kind of going along a DNA molecule, it's looking for a special promoter to start the process of transcribing. And that promoter is known as a Tata box. And it's called a Tata box because these letters, TA, TA, refer to the base sequences that make up this box. It's thymine, adenine, thymine, adenine. And so here is a sample of what a DNA molecule can look like on both sides. And so you'll notice here that you have this Tata box, which acts as a promoter for an enzyme. And the enzyme that we're going to be dealing with, let's make it orange, is going to be known as, let's make this bigger too, is going to be known as RNA polymerase. RNA polymerase. If you have just watched the video on DNA replication, you'll know that a polymerase enzyme is used to build new polymers. In this particular case, we are building a polymer of RNA. And it's a specific type of RNA known as mRNA. So it builds mRNA or messenger RNA. And that's going to be like our little piece of paper here that can then leave the nucleus. Imagine here that after this RNA polymerase finds the Tata box, it actually separates these strands, and it's only really going to be reading off of one side of them. So it's going to be reading here, and it's going to be making a short copy of the DNA, but as complementary side and RNA. So here our T is going to be an A. And then remember, there's no thymine in RNA, so this A is going to be a uracil, a U. And so on, all the way up this molecule. Okay. And at this point, we're actually going to be coming to a section here. This AAUAA. This is important because once the RNA polymerase reaches this section here, this indicates where it's known as a polyadenylation signal. And this kind of indicates to the RNA polymerase that it should stop transcribing. It doesn't stop right away. It continues for another, a couple dozen bases, but this is essentially the end of where the transcription occurs. So we now have our mRNA like this, and the mRNA kind of breaks free and can then leave the nucleus. There are some additional processes that happen before it does leave the nucleus. For example, on the 5' end of the molecule, you end up with a cap. It's known as a methylated cap because it's a methyl group. And on the other side, on the 3' end, you stick on a whole bunch of adenines, and it's known as a poly A tail. And this kind of just protects the mRNA as it leaves the nucleus and travels out of the nucleus into the cytoplasm. So here's our newly methylated mRNA leaving our nucleus. Here's our nucleus. And it's going out into the cytoplasm of the cell. And it has to go to the site of translation. And you'll remember from our little cookie analogy up here, this is a ribosome. All right, this is where the translation occurs. So this can either happen in a ribosome that's free in the cytoplasm or in a ribosome that's bound to a rough endoplasmic reticulum. But this is where translation is going to occur. All right, so if we zoom in on one of these ribosomes, we can see that it's actually made of some subunits here, and I've drawn them here, and we'll get into what all of this means. Specifically, it's made up of a large subunit known as a 60S subunit and a smaller one known as a 40S subunit. And they kind of come together like a hamburger bun and sandwich our mRNA molecule. And while it's inside of the ribosome, by the way, ribosomes are another type of RNA. It's known as rRNA. It's made of RNA, ribosomal RNA. Not super necessary to know at this moment. Let's keep that there. But as the ribosome kind of goes along the RNA molecule, the mRNA, there are sequences of bases that the ribosome can read. Specifically, it reads them in groups of 3, okay, of 3 bases. And every group of 3 bases is known as a codon, because these 3 bases are going to correspond to a particular amino acid. And you'll remember this whole process is for making proteins, so we want to string together some amino acids. So up here, we have this funny looking molecule that looks like a T, and that's why it's called, well, it's not really why it's called, but you can think of it as tRNA. This is transfer RNA. And what the tRNA does is it matches up with these codons, with these mRNA codons, and it starts to string together some amino acids. Every tRNA is carrying an amino acid, in this case methionine. And as it reads the different codons, different amino acids are going to get strung together. So after this one matches up, the tRNA breaks away, and the mRNA kind of pushes into the ribosome, and the next one comes in. And you'll notice this AAU section here is going to match with this UUA. This, by the way, is known as an anticodon. So tRNA molecules have anticodons, mRNA molecules have codons, and they pair together. Our tRNA is carrying a leucine here, and that's going to be the amino acid that is going to be represented by this UUA codon. I know this is a lot, but it's going to keep going until it reaches a particular codon known as a stop codon. And the stop codon releases, it's a release factor that releases the mRNA from the ribosome, and we have our polypeptide chain here. And so here is our polypeptide chain, and if you remember, this polypeptide chain will eventually fold up into a specific shape, and this is going to be our protein. Okay. Now, how do you know which amino acids are going to pair with which codons? We use this tool that's known as a codon wheel. There are codon tables, there are codon wheels. I got this one from Wikicommons. And what you do is you essentially read from the inside, from the 5' end of the molecule, out to the outside. We said that our first codon was AUG, and we're using this table here. We can see that starting in this section from the middle, this is our first circle, A, and then our second circle we have U here, and then AUG, well, that is methionine, and that's exactly which amino acid came first up here. In fact, methionine is going to be the start of all of our polypeptide chains. So methionine is going to be our start, and it's going to keep going and string together more and more of these amino acids until we reach a stop codon. And you'll see over here, the stop codon is represented by this black circle. So for example, if I reached a UGA codon, then that would release the amino acid. That would release the mRNA from the ribosome. Anyway, that is protein synthesis. I know it's a lot. Feel free to watch it again. Or if you have some questions, let me know in the comments and I can do my best to answer them. Thanks.